We're now going to look at part 3 of week 1 overview of the human genome. We will start off by looking at the mitochondria. These are organelles that provide cellular energy in the form of ATP to the cell. Remember that the more mitochondria a cell has, the more energy that is being produced. They contain a small circular DNA called mitochondrial DNA or mtDNA, consisting of 37 genes. There's no crossing over and little DNA repair. There is a high exposure to free radicals due to the production of ATP. The mutation rate is greater than it is in the nuclear DNA, and they exhibit exclusively maternal inheritance. Here you see a picture of a mitochondrium, and you can see the double layer membrane. And this double layer membrane has invaginations on the inside called cristae, which help to increase the surface area there so that they can perform the oxidative phosphorylation to produce the ATP. The enzymes are found on the cristae are called oxysomes, and they have a lollipop-like structure to them. If you can remember the endosymbiotic theory about chloroplasts and mitochondria actually being bacteria that were engulfed by a host cell, this makes a lot of sense because of the fact that the mitochondria are able to self-replicate. They replicate on their own. They do not require the host to replicate. The cell typically has thousands of mitochondria, and each has numerous copies of its many chromosomes. Here you see a pedigree, and in this pedigree you can see that there's maternal inheritance. You can see here that the father does not pass on any genes. It is the mother that passes on these mitochondrial genes, and that is because, if you remember, the mitochondria are found in the cytoplasm, and when the egg and the sperm fuse during fertilization, it is only the nucleus of the sperm that contains the DNA which fuses with the egg, so there is no cytoplasm involved. The condition called heteroplasmy is a condition where the mitochondrial DNA sequence is not the same in all copies of the genome. The mitochondrium will have different alleles for the same genes. At each cell division, the mitochondria are distributed at random into daughter cells. If an oocyte is heteroplasmic, it has a differing number of copies of mutant mitochondrial DNA that may be transmitted, and the phenotype reflects the proportion of mitochondria bearing the actual mutation. And here you can see a slide showing an oocyte where the mitochondrium has a mutation and where it does not have a mutation, and what can take place during fertilization and then in development. So in 1991, two amateur historians found a grave that they believed to be that of Tsar Nicholas and his family, who were murdered in Russia. The remains of the suspected Tsar were then taken and compared with his living great-grandniece Xenia, and it was found that they differed at nucleotide position 16169 in the mitochondrial DNA. This was retested, and the remains then shown of the supposed Tsar, that for this site in the mitochondrial genome, the Tsar had in some samples thymine, and in others he had cytosine. Then the bone fragments were taken from a Grand Duke Giorgio Romanov, another relative, was dug up in 1994, and it was found by looking at his bone cells that the Grand Duke had either thymine or cytosine at the nucleotide position 16169. Therefore, the Romanov family had heteroplasmy. When you look at what the role is of the mitochondria, they can provide a powerful forensic tool, and they can be used to link suspects to crimes. They can identify war dead. They can support or challenge historical records. For example, it was believed that this grave where our little boy's body was found was that of the Dauphin, who was the son of Marie Antoinette and Louis XVI. And by looking at the mitochondria and then comparing the DNA and comparing it to some mitochondrial DNA from Marie Antoinette, it was found that this indeed was the son, the Dauphin. The mitochondrial DNA is likely to survive extensive damage, and cells have many copies of it. And researchers consult mitochondrial DNA sequences in order to reconstruct ancient migration patterns. So let's look at the organization of the genome. Chromosomes are not just a random collection of different types of genes and other DNA sequences. Regions of the genome with similar characteristics tend to be clustered together, and the functional organization and the genome reflects its structural organization and sequence. Some chromosome regions or whole chromosomes are high in gene content. They are said to be gene-rich, while other chromosome regions are low in gene content, and they are gene-poor. Abnormalities of gene-rich chromosomes tend to be much more severe clinically than similar-sized defects involving gene-poor parts of the genome or chromosomal regions. If we look at single copy DNA sequences or unique DNA, they make up about a half of the DNA in the genome, and most of their function is not known. But repetitive DNA sequences are in several different categories. One useful distinguishing feature is whether the repeated sequences, the repeats, are clustered in one or a few locations, or do they consist of arrays of various short repeats organized in tandem in a head-to-tail fashion. They are interspersed with the single copy sequences along a chromosome. The clustered repetitive sequences make up about 10% to 15% of the genome. The tandem repeats are collectively called satellite DNAs. In looking at repetitive DNA and disease, both ALU and line sequences have been implicated as the cause of mutations in hereditary disease. Addition type of repetitive DNA is found in many different locations around the genome, including sequences that are duplicated. When duplicated regions contain genes, the genomic rearrangement involving the duplicated sequences can result in the deletion of the region and the genes between copies, and this gives rise to disease.
If we look at epigenetics, it's the study of heritable changes in cellular function or gene expression that can be transmitted from cell to cell and even generation to generation as a result of chromatin-based molecular signals. These changes include modifications to the DNA, such as DNA methylation. You also can have histone modifications that alter the chromatin packaging or access. You can have substitution of specialized histone variants that mark the chromatin associated with a particular sequence or regions in the genome. Epigenetic mechanisms do not alter the underlying DNA sequence. That is extremely important to remember, and this distinguishes them from genetic mechanisms which are sequence-based. Epigenetic marks and the DNA sequence make up the set of signals that guide the genome to express its genes at the right time, in the right place, and in the right amounts. The role of the epigenetic changes in human disease in response to environmental or lifestyle influences is what is so exciting today in the world of epigenetics. The epigenome learns from experiences. Remember, the DNA code is fixed, and the epigenome adjusts to rapidly changing environment. Things you can change: stress, your nutrition. Looking at DNA methylation, it involves the modification of cytosine bases by methylation of the carbon at the 5th position in the pyrimidine ring. Typically, it inhibits gene expression. So when you think of DNA methylation, you want to think typically, that it inhibits the gene expression. Histone modifications consists of modifications to any of the core histone types, H2A, H2B, H3, and H4. It includes histone methylation, phosphorylation, acetylation, and others at specific amino acid residues. These epigenetic modifications are believed to influence gene expression. The ENCODE project examined 12 of the most common modifications and found that much more of the genome plays a role directly or indirectly in determining the varied patterns of gene expression that distinguish cell types. Here on this slide, what you can see are some of the epigenetic mechanisms, such as development, environmental chemicals, drugs, aging, diet, and what they can do by way of methylation and histone modification, so that at the end point you have cancer, autoimmune disease, mental disorders, and diabetes. I would like you to look at a exercise that was put out by the University of Utah involving nutrition and epigenetics. They have some nice pictures in there and some great films. So I want you to look at that, and I want you to do the nutrition exercise, and then I would like you to answer the discussion questions at the end, and then you will respond to your classmates. You must have two, at least respond to two of your classmates after answering the two questions. And I'm not going to say much more about the nutrition and how it's affected because we will discuss it. And I want you to look at this exercise first. I want you to look at the epigenetics um, film on twins. I want you to look at this. It's very important, and you will also be looking about BPA with the plastics, and there will be some studies with rats. The process of genomic imprinting involves the silencing of either the maternal or paternal copy of a gene during early development. It's believed to be caused by methylation. We will be looking at diseases that are caused by genomic imprinting. And we will begin to start to look at what are the basic causes of the different diseases that individuals have. We'll start to look at that next week.